Free radicals are highly reactive species that are present in our bodies. The most important types present in our body are those that are known as reactive oxygen species. Reactive oxygen species are simply chemically reactive molecules containing oxygen. Research suggests that free radicals play a key role in both normal biological function and in certain human diseases. Free radicals and other reactive oxygen species are produced in all living organisms and have biological advantage. They are essential in the synthesis of energy and essential molecules, and they are also involved in boosting our immune system.
When free radicals and other reactive oxygen species accumulate in the body, they cause damage to cells, DNA, lipids, sugars, and proteins. The damage caused by free radicals and reactive oxygen species in plants and animals could lead to deterioration of foods, cell membrane dysfunction, protein modification, enzyme inactivation, and the breaking up of DNA strands. Free radical-induced oxidative damage may be a precursor to aging and diseases such as
something
cancer.
like this. And as you can see, this is a calcium graphene sandwich, and basically what it does is it allows the electrons to move more freely due to the properties of calcium and graphene turn into a superconductor. But then again, they still rely on the fact that they had to turn the temperature way down for the effect to appear, or about 2 to 4 kelvins to be more precise. For those of you that might not know, the best online source of knowledge, Wikipedia, generally defines superconductors as: it is a phenomenon of exactly zero electrical resistance and expulsion of magnetic flux fields occurring in a certain materials, called superconductors, when cooled below a characteristical critical temperature. Essentially, for you to reach that state, you have to use certain materials at nearly 0 Kelvin, which makes it impossible in everyday use. And for the longest time, scientists have been looking for other ways to achieve this phenomenon with many compounds, including but not limited to.
Viruses are not living things. Viruses are complicated assemblies of molecules, including proteins, nucleic acids, lipids, and carbohydrates. But on their own they can do nothing until they enter a living cell. Without cells, viruses would not be able to multiply. Therefore, viruses are not living things.
Normally, oxygen's got the 4 pairs of electrons here, but in a free radical situation, it might gain an extra electron. So now you've got this one unpaired electron, and we call this guy a free radical. And it's what causes all the trouble and potentially can cause cellular injury. Now, free radicals are generated both normally in physiologic conditions and abnormally in pathological conditions. Physiologically, so we're talking like normal conditions, it can happen during oxidative phosphorylation, which we know is this super important process in our cells to help make ATP. During this process, you've got this molecule called cytochrome C oxidase, or sometimes it's just known as complex 4. This molecule transfers electrons to oxygen, and oxygen gladly accepts those electrons. And this in turn causes the mitochondrial matrix to pump protons out. And this creates our proton gradient and drives the production of ATP. Great. So normally, for oxygen to be happy, it accepts 4 electrons and becomes water. But what if it doesn't receive all 4? Well, that, my friends, is when we get free radicals. Okay, so if oxygen grabs just 1 electron, it becomes superoxide. So O2 with a little dot for its extra electron. If it gets 2 electrons, it becomes hydrogen peroxide. And then 3 electrons, it's the hydroxyl radical. Then finally, if you get all 4, you get water. So 1, 2, or 3 electrons is called a partial reduction of oxygen. So oxygen isn't being reduced all the way to water. Because remember that reduction is a gain of electrons. So in this case, it's only partial. And remember that this happens physiologically, like in normal conditions. Also, though, you can generate free radicals in pathological conditions. So not in normal conditions, right? One way is through ionizing radiation, and this usually generates a hydroxyl radical. And this will happen when the radiation sort of comes in and hits water in your tissue. So it hits the H2O, right? And it knocks off an electron. So if you go backwards here from water, you'll end up at the hydroxyl radical, right? And these hydroxyl free radicals are generally the most reactive, and so therefore probably the most damaging to your cells. Besides ionizing radiation, though, inflammation is another way that you can get free radical generation. When you get an infection and neutrophils come by to fight that infection, there are two mechanisms they use to kill whatever microbe there is, one of which is oxygen dependent. Oxygen dependent means you need oxygen, so it can probably guess that oxygen's involved. Usually this starts with what's called an oxidative burst, where an enzyme called NADPH oxidase takes your oxygen and converts it to superoxide. And then it goes on to eventually be converted into bleach, or HOCl. But mainly our culprit for producing free radicals is going to be this first step, where NADPH converts oxygen to superoxide. And a third way that we can produce free radicals is through contact with metals. And we're usually talking about metals in the context of the body, so like copper or iron, where they're usually bound to something. One example are something called transferrins. These proteins bind iron and sort of help you control the amount of iron in your blood. Why do we want to make sure it's controlled? Well, it's because if it's not bound, if it's not controlled, it can generate free radicals. A reaction called the Fenton reaction allows iron to generate a free radical, usually the hydroxyl free radical, our most dangerous one. So let's think about it in the context of some disease, like hemochromatosis, where iron builds up and you get serious tissue damage, like cirrhosis in the liver. The primary mechanism behind this is a buildup of free radicals. Similarly, in Wilson's disease, there's this excess of free copper, so copper that's not bound. In the same way, you end up getting this tissue damage from free radicals. Okay, finally, there are drugs and chemicals that can also produce free radicals. For example, a drug like acetaminophen, which, like many other drugs, goes to the liver to be metabolized. In the liver, the P450 system takes care of it, which is like this group of really important drug metabolizing enzymes. This metabolizing, though, can generate free radicals, and so when high doses of acetaminophen are taken, it can cause massive death of tissue in the liver, and this is mainly from free radical damage. Okay, so all this talk about free radicals causing damage, but how do they actually damage the cell? Well, one big way is called lipid peroxidation, meaning that they can sort of steal an electron from the lipids of cell membranes. Remember that they have this unpaired electron, so they want another electron to be its pair. So what can happen is it can take an electron from a lipid in the cell membrane, which leaves that lipid with an unpaired electron itself. And then that lipid, now that it has an unpaired electron, can do the same thing to another lipid, and it can propagate this process in sort of a chain reaction. And as you can imagine, this can hurt the cell membrane and damage the cell as a whole. Free radicals can also oxidize both proteins and DNA inside the cell as well. Oxidation of proteins can obviously hurt the cell, depending on the protein's function, but oxidation of DNA is serious and is a super big player in oncogenesis, or causing cancers, since cancers are formed by mutations of DNA. So if these free radicals oxidize DNA and introduce mutations, all of a sudden your risk for developing a cancer goes up. Okay, so since this can happen in both pathologic and physiologic...
The head of a pin is 1 millimeter across. Divide by 10, 0.1 of a millimeter, the width of a human hair. Divide that millimeter by 100 and you get to the size of ultrafine water droplets found in fog. The smallest bacteria, hundreds of nanometers wide. A strand of DNA inside that bacterium, which houses its genetic code, is about 10 nanometers across, and a nanometer itself is less than 10 carbon atoms long. Nanotechnology is about the behavior of materials or devices at these incredibly small nanoscales. Technology somewhere between bacteria and atoms. You know how big bits of material behave from years of science, engineering and everyday experience. But at the nanoscale, tiny bits of exactly the same material don't behave the way we would expect at all. Take gold, or as nanotechnologists call the gold we're used to, bulk gold. Dense, soft and shiny. It's the most ductile, stretchable, and malleable, flattenable, pure metal known. It's inert in the human body, it's a great conductor of electricity, it has that attractive, bright, yellow, well, golden colour. But nanogold, the tiny particles of tens to thousands of gold atoms, is none of those things. It's not inert. Depending on the particle size, it can be very reactive or a very good catalyst. It's not a conductor, but a semiconductor. It appears red, or black, or dirty yellow, depending on the size and shape of the particles, but never gold. Why not? Bulk gold. Its colour, like that of any object, is a complex combination of how incoming light is bounced off, passed through, or absorbed. It absorbs the blue light, and the rest of the spectrum, the reds and the greens, are reflected and combined to make up gold's characteristic yellow colour. So far so good. But we need to dig a little deeper to understand why nanogold behaves differently. While it's convenient to think of gold as a hard wall of atoms, the science is a little weirder. It's not a wall, but rather a shared sea of loosely bound electrons that slosh back and forth over the bulk gold surface at regular intervals. It's like the slosh of water you can set up by sliding back and forth in a bathtub. The frequency of this wave determines what light is reflected and what light is absorbed. The name for this rate of sloshing is the surface plasmon resonance. Brilliant name. Sounds like something out of Doctor Who. Nanoparticles are far smaller than bulk gold, so their shared electron sea is more like a pond. The nanogold pond sloshes back and forth faster than the bulk gold sea, and the frequency of light it absorbs is therefore different. It absorbs yellow and blue light and reflects, you guessed it, red light. And for different nanoparticle sizes, the surface plasmon resonance changes, and so does the colour of the nanoparticle. So bulk gold and nanogold are both gold, and therefore made up of gold atoms. But the far smaller particles in the nanogold result in different physical properties, such as having a different colour. Nanotechnologists can choose a particle size not just for gold, but for any material. This allows them to test, fine-tune and retest nanoparticle properties in the search for new solutions to a whole range of problems.